Good. Okay, so thank you very much for the introduction. Um, as was mentioned, I'm studying the fate and behavior of diluted bitumen. This is work done with Dr. Jason Ahad in the Delta Lab at INRS in Quebec City, as well with close collaboration with Dr. Richard Martel, who is a contaminant hydrogeologist at INRS, a professor. And as well, I'm supervised by Dr. Valerie Langlois, who is an ecotoxicologist, also a professor at INRS Research University in Quebec City. So just to give a quick introduction to my talk, I'll define what is diluted bitumen, or as it's referred to as dilbit. So bitumen is it's an unconventional oil found in very large reserves in Western Canada. It's close to the surface relative to other oil types, and so it's been exposed to wind, water, solar, different forms of degradation. So it's a fairly viscous type of oil. And because it's so viscous, it's very difficult to manipulate and very difficult to transport. So it's often diluted with diluents, often naphtha-based condensates. You could think about 70 to 80% of that would be bitumen, and about 20 to 30% would be naphtha-based condensates. And that's why it's called diluted bitumen, or for a shorter form, dilbit. It's predicted that up until the year 2035 or even beyond that, Canada will be increasing its overall production and transportation of dilbit. And that's been increasing for the last 50 years and will continue to do so despite transitions to more carbon neutral forms of energy production. With more bitumen being transported, more of this dilbit being shipped via pipelines, rail, truck, we have a greater risk of spill events. The largest two inland spills of oil within North America have both been diluted bitumen spills. The most recent of which was in Mill Creek in Kansas in 2022. This spill was about 2 million liters of oil. This was a fairly recent spill event in December of 2022. So the extent of environmental contamination and the reports and the analyses that are coming out of that have not yet reached public surface yet, so it's not quite publicly accessible. So we're going to see what those results will be, but it's been quite a number of years since the Kalamazoo River spill in 2010 in Michigan. This spill was about 1 million liters from a ruptured pipeline. This spill was, of course, significant not only because of the catastrophic effects in the environment, as well as the risks to human health and safety, but as well, it was determined from this event that the environmental fate and behavior of dilbit when it's spilled is not as predictable at the time as conventional crude oils. And by that, I mean, when that oil was spilled and was contaminating the Kalamazoo River system, they utilized conventional crude oil collection techniques. So they put booms out on the surface of that water to collect the oil that was floating along the surface. However, that diluted bitumen sank within the water column, which was unpredicted at the time and caused more extensive environmental contamination than might have been predicted otherwise. And so it became very expensive to clean up this operation, as well as it caused greater contamination, greater risk to human health and safety, as well as natural resources. So there was quite a bit of interest by the federal governments of Canada and the United States to get a better understanding of that environmental fate and behavior should there be spills in the future. So this created the opportunity for our research projects. And we have some of the objectives of our research listed here. This is not the entirety of my PhD, and unfortunately, it's difficult to get a PhD down in 200 pages, but just to get it in 15 minutes is, of course, going to be difficult in itself. So I have to limit all this to kind of generalized information and just try and highlight the main points. But if you do have questions, of course, you can always contact me after, and I'd be happy to discuss that. But for the objectives that I'll talk about in today's presentation, I'm going to stick to two different spill scenarios that we tried to model. So in the laboratory, we utilized mesocosm systems, and we focused on terrestrial systems where we spilled on the surface of an environment in soils. And that we wanted to assess whether that would contaminate groundwater systems below that surface. And as well, we assessed surface water spills, so spilling in an environment that would mimic a freshwater lake or river environment. And then the second and third objectives were to look at the environmental fate and behavior through looking at the evolution of the chemistry of that oil over time in its transport, as well as looking at the subsequent toxicity of that oil as it weathered over time. So for that first component of our studies, looking at terrestrial spills, we utilized large mesocosm column systems owned by Dr. Richard Martel. And these are systems utilized to mimic unsaturated anaerobic conditions within the vadose zone. So they're about 100 centimeters in height and 60 centimeters in diameter. And we filled these systems with 300 kilograms of soil. And then we added water to these systems, about 6.25 liters per week, to mimic precipitation at the site at which the soils were collected, which was the Mont Saint-Hilaire Nature Reserve, which is a UNESCO World Heritage Site, and also located quite close to the Enbridge 9 pipeline where diluted bitumen has been transported previously. So once that water had percolated through this system, we collected what came out at the bottom and analyzed that for various chemical and toxicologic analyses. We spilled 2 kilograms of oil onto the surface of those columns, as you can see depicted here. We had 2 columns, replicate columns for diluted bitumen, and we had 2 columns for a comparative conventional crude oil. So we wanted to compare between the two, the dilbit and the conventional oil. And then we had a column with a control soil, so no oil was added. And as I mentioned, we collected water samples and utilized those for toxicity analyses and chemical analyses. These are the results for toxicity analyses, specifically malformations. So we utilize the fathead minnow, which is an indicator species for freshwater environments in North America, where these pipelines currently traverse and transport diluted bitumen. We looked at malformations here. This is a 7-day-old larvae, which has no malformations. It has normal growth. And this would be the example of a malformed larvae. So it has malformations that are typical of petroleum exposure, which include various forms of edema, as well as spinal curvature and craniofacial malformations. Also, they would have reduced body size and impacts on the cardiovascular system and further on and so forth. But just for malformation purposes, this is the data that we're showing here. Each one of these data points represents an individual toxicity test. So these fish were exposed for 7 days and then collected. So each one of these data points represents one of those toxicity experiments. So that water was collected from the columns on this time point, this time point, this time point, and those are individual toxicity experiments. The red triangles are for the groundwater contaminated with dilbit, and the blue diamonds are for the groundwater contaminated with conventional crude oils. And then the controls are the white circles. What we observed from our experiments is that within the first 34 days post-spill, we see significantly greater malformations for those fish that were exposed to the groundwater contaminated with the dilbit and the conventional crude oil, whereby beyond those 34 days post-spill, that water was no longer causing malformations that were significantly greater than the control fish. We saw the greatest level of malformations 34 days post-spill, but I'll mention that there were no significant differences between the fish exposed to the diluted bitumen and the conventional crude oil. These are the results for CYP1A analyses. CYP1A, you can think of it as a very sensitive biomarker to petroleum exposure. In response to exposure to petroleum compounds at the cellular level, the cells will, for many living organisms, will produce CYP1A mRNA, which is involved in the detoxification process of petroleum compounds. So when you see elevated levels of CYP1A expression, you see an overall increase in the accumulation of petroleum compounds at the cellular level. So here we see increasing CYP1A expression in these organisms up until 34 days post-spill. And this matches quite closely to our malformation data. So we could suggest that indeed these malformations are very likely to have been induced by petroleum exposure. Again, no significant differences between the oil types, but we do see overall the greatest toxicologic effects up until 34 days post-oil spill. So when we look into the chemistry to try and understand what's causing these toxic effects, these are the results we have for the concentrations of monoaromatic compounds in the contaminated groundwater. So we saw some higher concentrations in the diluted bitumen contaminated water than the conventional crude oil, but that was reflective of the original concentrations of these monoaromatic BTEX compounds within the original source oils. We do see high concentrations that, according to the scientific literature, would likely have effects on mortality and malformations for these fish at this time point before 20 days post-spill. However, it does not explain why we saw the greater malformations 34 days post-spill for the fish that were exposed to that contaminated groundwater. We're looking at polycyclic aromatic compound concentrations here. Again, we see some increasing concentrations throughout the experiment, and they do continue to increase beyond 104 days post-oil spill. However, I will point out that these concentrations are nanograms per liter or parts per trillion, which is about 1,000-fold less than what is reported to cause malformations in the scientific literature for polycyclic aromatic compounds. So it's very unlikely that these PACs are causing toxicologic effects in regards to the malformation analyses that we conducted. And as well, it doesn't explain why we're seeing the greater toxicologic effects 34 days post-spill. These are results for acid extractable organics. So this includes the classical naphthenic acids, which are deemed to be the most toxic component of oil sands process affected waters. So when they're doing the processing of diluted bitumen, the leftover water that's emptied into large tailings ponds, that's the water that I'm speaking of. And the naphthenic acids are they're shown to be one of the most toxic components of that water, the naphthenic acids. So we wanted to look at that in this groundwater contamination to see if that was somehow correlated to the increased toxicity. We do see concentrations increasing over time, but once again, we don't see any correlations around day 34 that might explain why we're seeing that increased toxic effect at that time point. So this is quite concerning because these are the compounds which are most often assessed: the PACs, monoaromatics, naphthenic acids. We also looked at metals, other ion analyses, total organic and inorganic carbon, and we're not seeing anything that correlates with the observed toxicologic effects. However, when we break down those acid extractable organics fraction into species classes, we do observe that there's differences between the control groundwater and the diluted bitumen contaminated groundwater in regards to the presence of O2S and O3S species. Those concentrations increase in relative abundance in the dilbit contaminated groundwater, as well as for the conventional crude, which is not shown here, but it's very similar in its results. Particularly when we look at the data for the compounds that are present within the first 34 days post-spill, around 34 days post-spill, we see the increasing proportion of compounds present in that groundwater, which would have a carbon number size between 6 to 10 with no double bond equivalents. So it could be possible that there's sulfonic acids, for instance, that are present within this groundwater that are causing these toxicologic effects, but this is something that needs to be investigated further. So I'm going to quickly jump into the surface water experiments. Instead of columns, we utilized a large tank system with mechanical arm for wave action, shoreline, and sediments. So this mimicked a freshwater lake environment. We had about 1,200 liters of water and 10 liters of oil spilled into that. So we had one system with diluted bitumen and one with a conventional crude oil, a medium sweet blend, and that was weathered for 56 days. We look at the toxicity data. Overall, we saw indeed that the contaminated water was causing toxicologic effects. So it had significantly greater malformations, significantly smaller body sizes, lower heart rates for these fish. But across the board, we saw that the fish exposed to the conventional crude oil had significantly more toxic effects than the diluted bitumen. So this was a finding. However, when we looked at mortality, we saw that it was more toxic from the diluted bitumen. And up until 14 days post-spill, we saw significantly greater mortality for those fish exposed to the diluted bitumen contaminated water versus that of the conventional crude oil contaminated water. So sublethal endpoints, more toxic from the conventional crude oil, but for lethality, we saw more toxic effects from the diluted bitumen. We looked at the chemistry to try and understand that. We had very high concentrations of BTEX 1 day post-spill. However, those concentrations decreased quite rapidly and then remained quite negligible on day 14 and thereafter. So it's not explaining why we see that greater lethality for the diluted bitumen 14 days post-spill, but it's likely that it was contributing to lethality that 1 day post-spill. For polycyclic aromatic compounds, we saw overall, the conventional crude oil had higher concentrations than the diluted bitumen throughout the extent of the experiments. This likely explains why we saw greater toxicologic effects for sublethal endpoints like growth, malformations. These are very closely correlated with PAC concentrations in the scientific literature, but we don't see, again, evidence for why we're seeing the greater lethality up to 14 days post-spill in regards to PAC concentrations for BTECs or for PACs. For acid extractable organics, we saw an increase in the concentrations for dilbit, and this could correlate with lethality, but it doesn't deviate much throughout the experiment, so it doesn't necessarily explain why we see 14 days post-spill greater lethality. But again, when we break that down into acid extractable organics, we see that there's a much higher relative abundance of O2 species dominating the chemical composition of those AEOs, particularly within those first 14 days post-spill. So there might be some correlations there, and that's another avenue that needs to be addressed and investigated further. So just as a quick conclusion at this point, indeed, we do see toxic effects from these oil spills in terrestrial and aquatic environments, and it seems to be well correlated with the original oil itself and the chemical composition of those oils. So if there's more PACs, we see more sublethal endpoints, but there's still some questions regarding malformations and mortalities. We do see that there's some correlations with oxidized species, these acid extractable organics fraction causing, we see correlations between the acid extractable organics fraction and the toxic effects that we observe. However, pushing forward, there needs to be further investigation into this specific species classes that are causing these toxic effects, because if we don't know what they are, we don't have the capacity to measure those in contaminated groundwater, for instance, or freshwater resources otherwise. And so we cannot deem whether or not that poses threat to environmental systems or potentially human health and safety. So with that, I will close my very rapid presentation. If you have any questions, you can reach me by email. And of course, I will answer any questions that I can now.